So, hi everybody. I'm Nico Katsanis. Many of you have known me, and I have known you for many years. Some of you might not have heard of the work, and some of you might not know anything about me. So, I'll just I'll tell you very brief. I am working at Lurie Children's Hospital and at Northwestern University. I moved to Chicago about a year ago after spending a decade at Duke and before that another almost decade at Hopkins, and before that at Baylor College of Medicine. My first involvement with Bardet-Biedl syndrome was in 1997. My goodness, when we knew so little, and it was bizarre and exciting and alluring, I was 20-something-year-old back then, and I decided to invest my entire career on trying to understand this disorder, both for intellectual satisfaction and to understand how the human body works, but also from a personal perspective. I have become frustrated for many years about our inability to do anything to understand rare genetic diseases in general. And I thought, why not this one? Why not try this one? And use it as a torchbearer, as it were, to try to understand and devise treatments for many other things. So what I'm going to do during the next 4 or 5 hours, no, I'm just kidding. Bill, please don't get a heart attack. What I'll do is I'll try to spend 30 to 45 minutes. I want to talk a little bit about the genetics, a little bit of historical stuff to put everything into perspective, and especially since I imagine some of the families who might be watching might be relatively new to this. I know from having, well, you taught me actually over the years, the emotional journey from having a child with suspected BBS diagnosis, confirmed BBS diagnosis, come to accept it. So I know that this is a journey. And I'm going to talk a little bit about the genetics and where I think the genetics is headed. There's at least one, I'm biased here because it's work that we've been pursuing for a very long time, but I think there's at least one very important study coming out in the next few weeks in the literature about this. I'm going to talk about some neurogenetics that merit discussion about how we're going to approach diagnostics. And then I'm going to spend the last bits to update everybody on some cool things going on in the therapeutic arena, because it's finally getting there. I shall say, for those of you who've known me for a long time, and I've had the privilege of knowing you for a long time, that you know that I just don't BS. And I also take a very, very dim view. We've had some difficult conversations over the years, and this is what you do when you work with a community of people that you love. And we've had some very difficult discussions over the years about snake oil and overpromising and under-delivering. And please don't stay frustrated with me, because I will continue to try to underpromise and over-deliver, because that's just the way it rolls. Okay, so gene discovery, which is the first bit. So, like I've said before, when I've had the pleasure of being with you in person, I mean, you're my people, right? So I hate this. I really, really hate doing it like this. But it's better than nothing. We'll be together next year. In the meantime, please wear your mask, right? Sorry, I had to plug that in. But on the gene discovery, we're now 20 years and 2 months since the discovery of the first BBS gene. I was there, 2 o'clock in the morning. It all happened. One of 2 labs that discovered BBS6, Val Sheffel's lab being the other one. And good grief, wow, right? So back in the day, we had less than 1% of patients with Bardet-Biedl syndrome being able to become molecularly diagnosed because we had one gene, BBS6. At the moment, I'll be blunt, I have stopped counting, because we're in the 20s right now. We're at BBS 23, 24, 25, depending how you count them. The important things are these. One, we've done a terrific job at being able to increase the diagnostic yield. So I want to say, 70, 80% of people, especially of people who have European descent, will receive a molecular diagnosis of Bardet-Biedl syndrome. And that is really, really important. And I don't want to talk about it too much, because you know this better than I do, the diagnostic odysseys, but also access to social services because a molecular diagnosis has been secured. So I think that's very, very important. For example, once people were able to get a BBS diagnosis, I know because there's some overlap with autism spectrum disorder, they've been able to access state controlled social services for autism. These are really good things. But also knowing about being able to understand about family planning, I understand. Now, I know also there's going to be people, I imagine there's going to be people on this call who still do not have a diagnosis, and I appreciate your frustration about this. And this is where we are. If you imagine that this is a curve, this is where there's going to be asymptosis, probably, because every new BBS gene we find, we tend to find one or two new families in the world with mutations in that gene. So all the big stuff, BBS1, then BBS10, then BBS2, then BBS6, and so on, it's all done. And we, the community, have been able to push a lot of this testing into the clinical services, which means you don't have to rely on Nico's lab when we get around to getting a molecular test done for 2 years. It's not because we don't care about you. It's because this is not what we're built to do. We're built to discover stuff. And a lot of the molecular diagnostic services we'll be providing to essentially bridge a gap with a clinical need. But you don't want us to be spending our time sequencing BBS patients with families to diagnose. You want us to be spending our time to discover new stuff, because by discovering new stuff, we propagate the knowledge. By propagating the knowledge, we get a better handle on the disorder, and we move forward. So obviously, we have seen a big difference in the interaction that we have with the patient community now, because a lot of people, not everybody, nowhere near as I would like to see everybody have access to clinical services and clinical genetic services. This is not a reality in the United States, but this is not a problem unique to BBS. We have reached the wall where we can. If anybody is living in Canada watching us, I know several families are from Canada. I'm so jealous of the access to the services you have. I don't want to say anything more about this because I think people know. We do have to pay attention, though, to some other things. For individuals who happen not to be European or of Middle Eastern descent, because a lot of good work has happened there, we have very little understanding of what the disorder incidence is in other populations. So, for example, in other Asian populations, we just don't know what Bardet-Biedl syndrome looks like there. And this is important. In African Americans, I should say this, especially at a moment where the country here is perhaps lifting his head a little bit and saying, wait a second, what are we doing? It is important for us to understand that in total, in my lab, we have studied something like 700 white families and about 3 black families. Okay? Is this because the incidence of BBS in African Americans is very low? Well, it's possible. We don't know. Is it because African American individuals don't interact as well with the healthcare service and with the research establishment? That is possible too. But I think we need to open our eyes and our minds and our hearts and think about this problem. It's the same thing. We have modest information about the Indian subcontinent. 1.3 billion people, 1 out of 7 human beings, and we really don't know what BBS looks like. So I think from the diagnostic point of view, I think we have a long way to go from a social perspective, not so much from a scientific perspective. And I think that the work that the BBS patient associations are doing, both in the United States, in the United Kingdom, in France, elsewhere, are a terrific conduit to open up access to people elsewhere in the world to actually get there. And I really want to encourage the communities to pay attention to this, to make sure that people who might not have as much, not everybody has access to medical care, but more people have access to the internet, which means that we have an opportunity to serve as an educational conduit to other people. And I think it would be very good if we, the custodians of the knowledge for all these years, actually improve the way that we can deliver the information to other people. And I look forward to playing a very minor part and look at the leadership for the communities, the patient community to lead the way. Okay, so the second thing I want to talk about in terms of mutations is this, and that is also important about how we deliver the data. So as many of you will remember, we have DNA. DNA is made of 4 letters, A, C, G, T. And in many patients, the cause of Bardet-Biedl syndrome is a single misspelling, a misspelling in a single base pair. So, for example, I think if we're assuming that everybody's on European descent on this Zoom call or YouTube video, 25% of you will have, or your children will have a mutation called M390R in BBS1. It's one of the most common mutations. What is M390R? Is a mutation in which the residue that encodes for a little for amino acid methionine is actually mutated, and it's converged to an arginine, and that causes the disease. And I know it seems ridiculous. We each have about 3 billion of these letters in our genome, and just one change causes this catastrophe. But this is the way genetics works. But over the last few years, we've begun to appreciate that there's other mechanisms that can also cause Bardet-Biedl syndrome. So one of the ways is that rather than having a mistake that affects a single base in the genome, you actually have a mistake that takes out a chunk of a chromosome. And sometimes it takes out a whole BBS gene, sometimes it takes a fragment of a BBS gene. Why is this important? This is important because the method by which we detect mutations may or may not detect these events. So, in a classical way, when we send for a molecular genetic test for Bardet-Biedl syndrome, it used to be a gene panel test, or it used to be what some of you will know as a whole exome sequencing test. These tests may or may not have the sensitivity to detect these larger deletions. I know it sounds counterintuitive in that these tests have the sensitivity to catch one single misspelling, but they cannot catch. So if you think about it like a book, right? If the genome is a book, think about it like there could be a whole page missing, and these tests can't see that, but they can see a single misspelling of a word, and the answer is yes. That's exactly the way it works. And the reason is this. The reason is what they're comparing against. They're not looking at the book to see what's missing. They're comparing this to another book. And most tests, if what is missing is small, they don't have the sensitivity to catch that. Now, as technologies move forward, we will be able to address that as well. If the piece that is missing is very large, for example, in a gene called NPHP1, you're missing 220,000 letters, all in one block. That standard clinical testing will catch. And many of the sort of molecular tools that many of us use will catch very easily. However, we now know that as much as 10 to 15% of patients with Bardet-Biedl syndrome, we will have changes where they're missing 100 letters, or 50 letters, or 30 letters around a BBS gene. And when that happens, a lot of the classical tools we use for mutation detection will fail. So this is very important, because obviously for accurate diagnostics, I suspect that we might actually be able to pinch on this 70, 80% and get it a little bit better to deliver diagnostics on a few more people. But the other thing is that the thing I'm going to talk about next, which is the idea of genetic burden. On top of that, as we become better and better at all this, now that we have all these genes and we know where they are in the genome and we know how they're arranged in the genome, so genes also have another component, which is bits of the genome that tells the genes when to switch on and when to switch off. So think about it as a stereo, right? You have your components that make the music, but you also need some sort of controller to tell you on, off, and by how much, like a wrist. And it is reasonable, it has been reasonable to imagine that if we actually have changes in those elements of the genome that affect the function of the BBS genes, this will also be involved in Bardet-Biedl syndrome, and that is actually turning out to be true. These things are much harder to detect. These things are much harder to validate, but they're beginning to come through. We played a very small part on a paper that was submitted about 48 hours ago that actually showed that there was a little element that was inserting itself near a BBS gene that was causing the gene to shut down. And this element is found in something like 4% of BBS patients, or some number, I forget. It's non-trivial number. So it's like everything else. I think I said a few years ago when we were all in Durham, I said to people that genetic data should be reanalyzed all the time, because as our knowledge base increases, our ability to interpret data improves. And it will always improve. And so, although the sequencing of a genome might be once and done, because your genome for the most part doesn't change since the sperm and egg met till your last day, your genome for the most part will stay fixed. It will accumulate some stuff, but for the most part will stay fixed. The interpretation of what we find changes heavily because genetics and genomics is a rapidly evolving field. And the other thing to bear in mind is that people will also make mistakes, because one day you interpret this way, the other day you interpret that way. The way to think about this is like the way, one of the strengths in the United States is the way the law is written and interpreted, right? It's a living document. It's not fixed in stone. Well, our interpretation of the genome is a little bit like interpreting the law sometimes. Because as our understanding changes, then our interpretation changes. Okay. So with that, I really want to segue into my second piece. So, sorry, just that. So, obviously we care about diagnostics for the reasons that everybody understands. And we will continue to pursue those. And for those of you who have not enrolled in a research sequencing study, please come talk to me immediately. I'm easy to find. We will need a pint of blood. No, we will not. We will need about this much blood. But dads, your children are braver than you are, because it's always the dads who are afraid of the needle, all right? So I'm going to put it out there. The kids are brave. The moms are doing okay. We dads, we are not tough. Some of you might be. I'm not. Yeah, we will probably need a little bit of blood and potentially a haluki, and then we can do some genetics. Because we will continue to pursue the modification of genes associated with Prader-Willi syndrome. Okay, now what is more interesting to me, because it potentially adds more value both to, well, the body of knowledge of the human race. I'm sorry, I'm not trying to be melodramatic here, but those of you who have had the chance to interact with me socially, know that I actually believe this stuff, right? I believe that we do this to improve the race, to improve outcomes, to improve understanding, to further knowledge. And I know that, I said this before, and I will say this a million times again, many of you have joined this journey not out of selfish reasons, but out of altruism. And I just want everybody to just remember that for a moment, because I think, I get emotional even over Zoom, right? But I think what we do speaks to the best of us, to the best about us as people, especially at a time in the country, in the world, where we are living in some very difficult conditions. So, irrespective of what your belief system is, I do want to invite everybody to think that the fact that we have all agreed to work on something and support one another through the foundations and all these things, it's not because we know that the probability that our own families will benefit is very small. And for some families who have been in this journey for 30, 40 years, none. And yet people still participate. And I consider this the greatest gift, because it constantly reminds us that we all aspire to something greater than ourselves. And I think that's all that matters. Okay. I'm going to step off the soapbox now. I really want to talk about variable clinical expressivity, because that is something that is going to become very, very important moving forward. So, as many of you know, especially those of you who have multiple children with Prader-Willi syndrome, or who within your social circle interact with other families with children with variability syndrome, you will know that the spectrum of clinical need is massive. We have anywhere from the weight regulation to the vision to the renal function to even how many extra digits you got from, no extra digits to 1 to 2 to 4. And this is something that has really vexed me for a long time. And I'll be blunt, it also kind of fascinates me because I find it interesting as an intellectual exercise. So I'm not going to just sit here and say, oh, it's all for you. No, this is interesting. Why are people different from one another? Something that is intensely interesting because we want to understand this. It is also important because as we started moving closer and closer and closer to doing clinical trials for various things about Bardet syndrome, we need to be thinking about natural histories and outcomes. And I know Bob Hawes and other people are thinking about this, which is great. Because if you have a variance that is this big and you're trying to correct it and to measure the correction to claim that you have success in a clinical trial, you can't do it. You've got to get to things that have low variance. So, we know now that a lot of the variance is just stochastic, right? Stuff happens. But we also know that a significant fraction of the variance in the way that children present with Bardet, how many features, how fast will the vision go? We've gone from really fast to not bad. And we have huge bias, right? Because when we see our own children losing their vision, we become that much more motivated to follow up clinically with an ophthalmologist and all this. We need to know these things. Renal function. It was the big unknown. I know Bob Hawes can speak for hours on this, but we didn't know there was a renal function defect in a lot of BBS patients, and now we do. Well, there is renal. I mean, it's again, renal function is 6 different things. Some people have immature kidneys. Some people have some filtration defects. Some people have other things. Who's got what and why? Why these things are important? First of all, we need to be able to predict some of these things, to be able to help guide people in their decision-making as they go through the clinical enterprise. Because, why see 20 doctors when you can see 5? And I know that a lot of the families have experienced a lot of frustration by going from specialist to specialist to specialist, who may or may not have seen a Bardet patient in the past. Now, having individuals who are well-versed in BBS and ciliopathy clinics and all these things, these are wonderful tools, and we are improving as a community. However, we do not know why is it that one patient is going to go into renal failure and another one will not. Would it be cool to know this? I've said this before and I will reiterate. Wouldn't it be cool to know, to be able to predict who is at higher risk of renal failure and use it as a metric to prioritize people who might benefit from transplants? Because we have very, very poor prognosis. And also, imagine a world in which you start having drugs for BBS. It's coming up. Well, I don't think there's going to be drugs for everything. I think there's going to be different drugs for different indications, which means we need to know during the course of 10, 20 years in the early lifespan of an individual BBS, what are going to be their major challenges? What are going to be their other risks that will affect quality of life beyond the obvious? And can we do something about these things? But we also have to be able to measure. So, for example, I know I'm getting the weeds here a little bit, but I think it's important for the families to consider these things. Imagine if a patient with Bardet syndrome is losing slowly their vision over a 15-year period. Okay. It happens. There will never be a clinical trial for that. We cannot possibly be dosing an individual with an unknown drug for 15 years and asking the question, will their vision erosion improve? We'll have to have a different measure. And there's ideas to this. But this is the point. So where do we stand with variability? Well, for many years, we have been making these observations that patients with Bardet syndrome tend to have an increased abundance of mutations. These mutations could be the single reading errors, one amino acid at a time in the BBS gene. It could be the big deletions that take out a bit of the gene. It could be other things. So we've been making these observations that patients with BBS tend to have more of those in addition to their genes. So it's interesting when I meet folks with BBS, they're like, oh, I'm BBS 1, I'm BBS 10. And I think we need to, I understand why people do this, and I understand the comfort of knowing what your enemy is. But I think it's just not, it's only half the picture, let's put it this way. So we recently completed a very large study, and many people participated, and we collected data from almost, oh my goodness, I don't know, 400, 500 BBS families, and maybe more. And the data are that if you are a patient, if you're an individual who has Bardet-Biedl syndrome, you have twice as much genetic changes in your BBS genes, all of them, than me. And that is really important. This is not random. So this has now been sort of studied to death, as it were. So that says, what does that say? That says that the mechanism of that this genetic disorder acts on people is as follows. You have your mutations that are inherited from parents, one from mom, one from dad. And that is essentially those of you who read literature will say this is the inciting event. This is the thing that will give rise to the disorder. But then you have this clouds of variation. Could be about 20% of BBS patients have at least one more mutation in another BBS gene. About 50% of BBS patients. I'm not going to go through all the numbers because this is not I don't want to give an academic lecture. I want to give people a sense of where we are. The point is, there is all this additional variation. It is significantly elevated above population. Now, we know this. Before it was episodes and people said, I see it, and then some other academician comes and says, I don't. Well, forget about the academic anecdotes because this is what they were. Now we have robust data because a whole bunch of academic labs got together and said, right, we're going to put all the data on the table and all we're going to do is count. So the experiment was very simple. We all know that to get Bardet-Biedl syndrome, you need 2 mutations. Right? So the question is, if you don't count these 2 mutations and you go ahead and you count every single change that you find in the BBS gene in a person with Bardet-Biedl syndrome, if the 2 mutations are all you need to get the disease, all the additional stuff should be no different than the general population. I hope that makes sense. I can repeat it one more time. If all you need to get Bardet-Biedl syndrome is the 2 mutations in a BBS gene, one from mom, one from dad, then the amount of genetic variation, mutations, that you have in the rest of the BBS genes should be no different than anybody else on the planet. Well, this actually turns out to be not true. What turns out to be true is that BBS patients have 2.4 times more genetic variation in the other BBS genes compared to the general population. What is even more important now, it gets more interesting. Even this variation is not randomly distributed across the 20-odd BBS genes, but it seems to be enriched across specific pairings of genes. So, for example, we know that about 50% of people of European descent will have mutations either in BBS 1 or in BBS 10. However, individuals with, BBS 4 is far more involved in genetic interactions with other things. We think there's some biology going on underneath. We just don't know what this biology is. What we are beginning to understand is this. As some of you will know, the BBS proteins aggregate we know of at least 2 major complexes. One of them is called the BBSome complex, and the other one is the chaperoning complex. Well, it turns out, it looks like the disease is going to be more severe and more aggressive if people have a mutation in the BBSome complex and a mutation in the chaperoning complex, as opposed to mutations only in the BBSome complex or only in the chaperoning complex. The way we interpret this data is that you have 2 complexes that are not working very well, so they actually interact with one another. Now, it's still early days for that piece of the work, but the point is we can already start thinking about stratifying individuals with Bardet-Biedl syndrome based on where the primary mutation is, what additional mutations there are, where do they map. and move into something that's called systems biology, which is to try to take all the information we have from this sort of bag of stuff that we carry and try to make better sense out of it. The one thing that is going to be key as we move forward is we need to get better at obtaining high quality natural history of the disease. And I know that we were making very good progress in that regard as well. And I'm very happy to say that because my group had absolutely nothing to do with that. So I'm very happy to give credit to others about this. But until fairly recently, the clinical information that we had about families from BBS were highly episodic. Some of it is just a note scribbled on a piece of paper. As we do better now, and as we're beginning to image better and become more systematic with questionnaires and things that people have been responding to, we're just gonna get better and better and better and better at figuring out what is the clinical progression of people. Because now we're in a position that we can put the clinical progression of people against the human genetic data, not just only which gene is the one that started the whole malaise, but what other variation is there. And we're beginning to see some glimpses. For example, we have at least 3 sites now that we know that if people have a particular combination of genes, they're far more likely to develop renal disease. So at the moment, it's a little bit more than an anecdote. It's not quite an anecdote because we've replicated this a few times now. It's not ready for prime time, but I want to share with people what I'm thinking about, right? Because wouldn't it be awesome if we're at the point where we can look at a patient's genome and say, we predict with pretty high confidence that this person might end up with end-stage renal disease. We need to monitor this very, very carefully. So this is where we're going to go. All right. So, so the last, and there's a lot of work going on there. The other thing I want to say is this is also going to inform, I know people might get a little bit nervous about this, so this is also going to inform who might be the most appropriate candidates for what clinical trial. So before anybody, I can already feel that's going to happen, all right? So let me be very clear, okay? We have to be, the most important thing is that we have a clinical trial that doesn't fail because we gave the wrong drug to the wrong people. That's so important. That's so important. At another time, on the next opportunity we're all together, I can share with you all the horror stories from the field. As you know, we run a small biotech company and we hear all these individuals here. But it is so difficult to develop a drug. You know this. Everybody knows this. It's a crying shame to develop the drug, test the drug in humans, and have it fail, not because the drug didn't work, but because imagine that you have a drug that will work in 1 out of 10 BBS patients. Imagine, I'm not saying that this is specific, but I want everybody to imagine this. I want to imagine that there's a drug that only works for people who have a specific mutation in BBS, 3. Please don't read anything into this. I just said BBS 3. BBS 23, okay? And we do a clinical trial where we include anybody who wants to participate. So we do 100 people, but only 2 of these 100 people are actually BBS 3. That clinical trial will fail because we will measure the aggregate efficacy of a drug across 100 people. And of course, that will fail. And our ability to initiate a second trial with this drug is horrendously difficult because getting more approvals from the Food and Drug Administration is going to be difficult, consents from universities, and also money. I don't talk about money in these things, but clinical trials will cost money. And if you fail, the motivation from people who give the money to pay for another one goes down because it's some people say there's at Einstein's definition of schizophrenia, right? You try again and again, and you expect to have a different outcome. So what we're hoping to do is to be able to understand which of our drugs, and I'll speak about the drugs in a moment, are going to be efficacious to which subset of BBS patients, with what mutational profile, and this is where we start. So my dream is we start with that, we see if there's efficacy, we see if people are getting better, we get FDA approval for that, And once we have that, we can extend to other groups of patients. And the regulatory bar and the cost bar and everything else is much, much lower. The key to BBS is showing, at least for some of the drugs that we have developed, is to show that the damn thing works, helps 3 people. If we can show that, that's very hard to do. If we can show that, then we can go and try to say, how about those other 5 people there, and those 10 people over there, and those 100 people over there, and expand like this. But we have to start very, very narrow. Okay. So for the last, I don't know, I think I have 15 minutes or so, I'm hoping to talk about, 10 minutes. I really want to talk about the drug discovery paradigm. I know there's gene therapy work going on, which is terrific. I know there is the peptide stuff, which I hope it works. I want to talk about small molecules. I want to talk about drugs, pills, injectable things. And the reason I want to talk about those is because I think that outside of a couple of very common mutations, BBS1 and 390R, C20FSX110, it's going to be very difficult to make argument for some of these other things. So the one of the things that is stunning challenge with variability syndrome is that so many genes, so many mutations. And in some ways, you have to treat each mutation in its own unique way. So what we've been trying to do is essentially go downstream of the mutation and treat the process. So to be blunt, I want a small molecule that I don't care if you have the BBS1 and 390R or a private mutation in BBS4 and you're the only person in the world who has it. I want to have a drug that protects your photoreceptors from dying. Because that's what it's all about. So, because if we go about who's got it and how many people, I mean, quite bluntly, for half the BBS genes, we'll never be able to run a clinical trial because there's only one family ever on the planet. And for them, it's going to be too late because by the time we've gone through all this rigmarole, for example, the photoreceptors are already gone. You can't bring them back. So by definition, that means that some of the work that's going on is only going to benefit recurrent mutations, BBS1, BBS2, and maybe a little BBS2. Okay, so where are we with this? A number of years ago, this is a long journey. Remember, this is why we don't talk BS, and this is why we speak the truth. A number of years ago, I told some of you, I hope you still engage with the community, that we made a number of observations that show that the mechanism by which the cell regulates the method by which it takes out the trash seems to be defective in patients with variability syndrome. And we suspected that this gave rise to at least some of the pathologies. We're particularly excited about the retinal degeneration aspect of the work because we think that this is an area where we might be able to do some good because we're catching it earlier now, because we have time to actually slow it down. So the dream is, of course, to slow down the loss of cells inside the retina to prolong people's vision, which will have major impact on many other things, language development, independent life skills. You know this much better than I do. Anything that I say will sound conceited. So we've been going after this pretty hard. At first, we had some stupid data, like there's a particular, I don't know if people remember this, but there was a particular compound that was found in broccoli sprouts or something like this. But the discussion was you needed to be eating something like, some kilograms of broccoli per day to actually get a little bit of that. So it was not tenable, but it was useful for us as scientists because it was called a tool compound to provide proof of concept. And the proof of concept worked very well. And that was good enough for us to go to invest the next period of time to do the next set of studies. For the next set of studies, what we did is we actually started asking the question, if I take a cell that is missing a BBS gene, and in this particular case, we started with BBS4 simply because we understand the behavior of BBS4 depleted cells better than no other reason. And for those of you who have, who are a person in your family with a BBS10 mutation, I have nothing against a particular gene. I love them all the same. It's just a matter of what mutations are more amenable to our tools in the lab to make a faster program. So what we did is we deleted, we took these cells and we deleted each gene in the genome on top of them one at a time. And we asked the question, what gene deletions might be beneficial? In other words, if I were to delete a second gene on top of a BBS gene, can I improve the outcomes in that cell? And what we discovered, I remember the day, it's clear as day now, shocking. What we discovered is that there were several proteins that were known to regulate this taking out the trash process, that if we were to delete them, things would go faster. All right. So we started a drug discovery program around those, and it became very clear very quickly that this particular protein cell made for chronic targets for a very simple reason, that you might restore retina function. And this is again why I remember, you know who you are. I remember people that, you gotta tell us what is this and all that. And like, I'm just not gonna do that because I don't have enough confidence yet to actually say anything. Remember, we do not bullshit. So, and I don't have an MD, so I don't have to be as cordial. I can speak. So it turned out that if you actually delete these proteins, it's just not the way to go for therapeutics for a couple of things. For a couple of reasons. First of all, they cause other problems. And for another thing, they're not always expressed in the right cells that we would like them to know. So for example, our two big targets, the eyeball doesn't make them. They're not present in the eyeball. So that wonderful therapeutic was not going to be helpful for the eyeball. All right. However, what we learned from that study was that we now had a mechanism inside the cell that we could measure. And we used that with the proteins that we discovered, and we ran a study on 2,500 small molecules. And the big deal here is that these 2,500 small molecules, the vast majority of them have already been approved for use in humans. Now, don't you all get too excited because I'm not going to tell you what it is, because that's how I roll. When I'm ready to say, I will say, because we need to be discussing who is going to be trying it. However, what happened is this. We identified 4 molecules that when we give them to the cells missing BBS genes, more than 1 BBS genes, they actually completely restore function. What happened next is many of you will remember that we have zebrafish models for many BBS genes. So in the zebrafish, a cell is a cell, I don't know what to study and how relevant it is, but in the zebrafish, we can study things like vision. So there's 2 experiments that are done there. The first experiment is you can actually take an animal and you can, you essentially cut through the retina and you look to see what the retina structure looks like and what the molecules that promote vision look like. Because we know from mouse models of Bardet syndrome and other studies that there's a difficulty in transporting molecules that cause vision in animals. Okay. The second thing is you can actually test if a fish can see. I know this sounds ridiculous, but you can. And the way you do this is you actually change. You have your fish swimming in a little space, and you alternate light and dark. If a fish cannot see, it makes no difference to them if it's light or dark. But when they can see, they react very specifically to light-dark transitions. And you can actually measure this. So this is something called an optokinetic test. And it's quite robust. So what we did is we took each of these 4 compounds and we did a couple of things. The first thing is that we looked at to see what is the dose. Again, Tylenol, right? If you take 1 pill, okay. If you take 20, not okay. So we need to know what is the dose and whether this dose could be tolerated. And it turns out the dose is fine for at least 2 of these 4 compounds. The dose is very, very good. It's something that humans will tolerate very well. So that's sort of hurdle number 1. Hurdle number 2, we actually delivered these compounds to the fish. We just injected them straight into the egg and then watched them develop. And what we observed, it was they prevented the retina from degenerating. So retina function looks robust. And the third thing that we did is this little rigmarole with the light-dark transitions. And in the light-dark transition test, those fish, these were BBS4 mutants, reacted identical to regular fish, not the mutants. So we have 2 drugs that have shown safety and they have shown efficacy in a cell-based model and in 1 zebrafish model. Is that enough to go to humans? Nope, so sorry. Is that good enough to go to other model organisms and test more? That's absolutely what's going on. So my hope and expectation is that, and the other thing to say is that primarily through the company, there is actually now a protocol where you can measure toxicity of drugs that will be acceptable by the Food and Drug Administration as part of the initial documentation that we'll need to provide to the FDA. So what we begin to think about now is that what are the data points that we will need to provide to the Food and Drug Administration to propose a clinical trial, and what will be the endpoints. So obviously, there's visual response to be one endpoint, but we do have a concern, and the concern is that it can take a very long time to see any improvement in vision. So we're thinking about other things, and we're thinking about muscle function, we're thinking about tone, obviously we're thinking about weight control, because that's an easy one. Many, many questions. So I do want people to get excited, because we are, but at the same time, for those of you who've known me for a while will know that I carry a very large grain of salt to anything that is getting very close to clinical studies, because we don't have that many opportunities to fail. So we're going to do, and the other thing is, I'll be blunt, I don't think I could live with myself if I delivered a clinical asset to BBS patients and something horrible happened to them. It's as simple as that. I understand, for those of you who met me personally, you know that my heart is with the community all the way. And the notion that every year that goes by and I can't deliver a drug is killing me. But, like I said to you before, and I will say this again, we, the community right now, represent a small piece of humanity in time. That what we do will matter to a very large piece of humanity in the future. And to be blunt, I think about 100,000 children who will be born with Bardet-Biedl syndrome in the next 1,000 years. And I want to make sure that I try to do something about that. And that is the most important thing. Because if we screw it up now, we might be pushing the field back another 10 or 20 years. So before everybody starts getting on the email to tell me what are the drugs and this and the other, I will not tell you. It doesn't matter what you do. I will not tell you. And that's not because I love you, because you know I do. It is because I respect the community and because I've got to do this the right way that I will not divulge information that I'm ready. I just want to share this progress because we are moving in the right direction, but we're moving in the right direction in a slow and methodical fashion, right? So please bear with us, and we will deliver the data when we think it's morally appropriate and scientifically acceptable to do so. The data will have to go through 13 rounds of peer review before I divulge anything. They'll have to be looked at by regulators and physicians and scientists, and we're going to poke them and probe them and try to find holes in the data before we actually say, because when we say, let's go, let's try this out, I think I want to be in a position where our probability of success is as high as it could possibly be. So, a lot to digest, I know. A lot to go through. For those of you who thought that the move from Durham to Chicago is going to be a little bit of downtime, you obviously don't know me very well. And for those of you who have championed the work for so many years, you remain in my thoughts and you will always have my gratitude.